Hello, we are Alan and Sasha, and today we're going to present our lab practice on the nervous system, where we conducted two experiments, which are the 2-point discrimination test and the reflex test. The first phase was a 2-point discrimination, where the goal was measuring the sensitivity in different areas of the skin. Our independent variable was the test area or dermatome. The dependent variable was the 2-point discrimination threshold measured in millimeters, and the control variable was the test subject. The toothpicks, the subject's eyes closed, and the pressure applied. We followed the procedure carefully by my partner closing their eyes while I applied 2 toothpicks in each of the 5 test areas, which were the upper back, middle palm of the hand, middle back of the hand, middle forearm, and the middle arm, staying very close together first, then gradually increasing the distance of the toothpicks until my partner felt 2 distinct points as one, and then we measured the distance with a ruler. In our results in both table and the graph, we can note how the sensitivity varies greatly, which shows that areas with more sensory receptors like the hands have a much finer sense of touch than those with lesser sensitivity, such as the upper back. Now, in phase 2, the reflex testing, here the dependent variable was the area of the body or the dermatome, while the dependent variable was our observation of whether the reflex response was positive or negative. Here the control variable is similar with the test subject, the position of the test subject, and the procedure of the checking of the reflexes. Since neither Alan or myself presented the reflexes, we resorted to have a friend be our test subject for this test. We tested 5 reflexes, which were the patellar, triceps, biceps, brachioradialis, and Achilles with the use of a neurological hammer. Yet, even though our test subject presented most of the reflexes, the biceps and the brachioradialis reflex wasn't present on him as well as on us. This shows how reflexes can vary between different individuals, as well as their neurological responses or reflexes. Then here is our reflex test table with the independent variable being the reflex test, dermatome, and also the observations of stimuli between negative and positive, which negative being the ones that wasn't present on the test subject and positive being the ones that were presented on the test subject. In conclusion, both of these experiments help us understand the sensory and motor pathways of the nervous system in a more visual way. The 2-point test shows us how different areas of the body have different field of stimuli, and the reflex test demonstrated how reflexes are different from each person and also how our spinal cord acts in involuntary movements. Thank you.